Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Thank you very much, Mike. Okay. Forgive me for putting my hand in the drawer. It's where I had to put the mouse. Right, okay, so what is Blast Atlas? Blast Atlas is a system that we're developing at QUT in our bioinformatics group. It builds on work that we have previously presented here, the Silver Map and Blast Explorer programs that Jim demonstrated in Pittsburgh last year. In a similar manner, it's focusing on Blast. That's the bioinformatics sequence similarity search tool, basic local alignment search tool. Okay. At a low micro level, perhaps, Blast Atlas is a visual analysis system for conducting genome-wide sequence similarity surveys. That is, it's aiming to help biologists who have sequenced a previously uncharacterized genome. They've identified putative genes in it. They may want to find out, A, if these putative genes are genes, or B, what they are. So one of the first steps they do is to throw those genes into sequence databases, get back lists of similar proteins, and then start to identify the likelihood that they're a gene, and the function of the gene, if it is one. So that's one clear biological use for what we're doing. Another more abstract and ambitious thing that we're doing is we're taking a sort of a step towards creating a virtual observatory for genomes. Now, the clear precedents are the Worldwide Telescope, Google Earth, Bing Maps 3D, that kind of system that has made it so that pretty well anybody can at least do something to observe the universe over the internet. You know, people can, in fact, they often use these tools not because they want to do anything scientific or geographical, but because it's actually just fun. You know, they enjoy it. Now, this is so I'm probably setting myself up for a big fall here, but that's where we're aiming with. This is a small step in that direction. So, bit of background. Since 2000, we've seen the sequencing of the human genome, and after that we saw the development of next-generation sequencing technologies, the new computational methods that were developed to enable the human genome to be finished in such short time. They've combined to allow us to now have access to literally thousands of sequenced genomes from organisms. NCBI reference sequence database, for example, contains over 1,800 genomes, and there's many more out there. Now, the data from these studies, raw genome data, is provided to the public via databases such as the National Centre for Biotechnology Information, EBI, and the DNA database of Japan. You know, you can go there. You can query their databases using web tools. You can download the GenBank files or the ASN.1 files. The data. So anyone with a web browser can get data. Also, there's sitting sort of at a slightly higher level, perhaps, we have integrative sort of synthesis collections. So the UCSC genome browser is founded upon 46 genomes from eukaryotes that are of scientific and economic importance. Okay, and then that's enriched by an extensive curated collection of multiple alignment data, cross-referencing positions on human to mouse, for example. Okay. In another aspect, there's an effort called Bio2RDF, which is aiming to use semantic web technologies to create a unified and interconnected map of all bioinformatic data that's available. So in a way, we've reached a point where we have free access to data. We can download stuff. We can query it on the web and do things. So we're pretty well in a similar position to where astronomy was in the year 2000. You could pull out data with FTP and you could grep your way through it and find all manner of stuff. Well, we don't use grep that often, but Perl and Python are still very standard tools. But in the 10 years that have passed, astronomers have got the Worldwide Telescope. Some of you probably saw the workshop this morning on it. I'm going to go to the one on Wednesday afternoon, but it's amazing. Google Earth and Bing Maps, these are based on a substrate of geospatial data, and with clever algorithms you can project that information out to users in their homes or in their cars or wherever, and it can be then overlaid with social, economic or scientific information. Now, people can get access to this stuff. They don't even know they're really doing a query. They just go there and they click. They might type in an address or right ascension and declination, perhaps. So what are we doing? We're trying to do the same sort of thing with genomes. One thing that we don't have that astronomers do and geographers do is we don't have coordinates. They're so lucky. It's a bit of a simplification, but given 4 coordinates plus or minus 20, a physicist can pin down most anything in the universe, and certainly you can get a 2-dimensional, a 3-dimensional, and a time-based reference to just about anything you need to talk about at a coarse scale. We don't really have that with genomes. The best we have is measures of this is similar to that, but there's no absolute where they are. But we would like to give scientists something that resembles these more advanced virtual observatories in that it'll be easy to use. They have a tangible, I won't say fun, but maybe user experience, and that they'll be able to see emergent patterns that you cannot see by looking at the data just with your eyes or by grepping or purling or whatever. So our approach is we start from some previously existing annotated and curated data, which has a defined biological purpose in the first place, and then we're going to try and build maps with it. So I think I've said enough about BLAST. So the starting point for the prototype I'm going to demonstrate today is a data collection called COG, Clusters of Orthologous Groups. Now, these are basically a collection of proteins derived from 66 representative bacterial genomes, which have been blasted against each other so that we have a similarity, or at least a statistical significance measure for how similar each pair of proteins in that collection is. And then they've been clustered after that based on initially E value, but that's the expectation value. And the clusters have been curated, and each cluster has been allocated to one or more of 25 functional groups. There's in eScience terms, it's kind of not an incredible volume of data. We're not getting petabytes coming off the line every minute, but this is a quantity of data that still exceeds the capacity of a human brain to certainly to hold, and also to browse even in a finite time. So the best we can do to process this at present is we do some variation of grep or awk. So here's a 2-dimensional map that we've produced, where each one of these blobs represents a single cluster in the COG database. We have 25 functions, and that's not part of the prototype. That's part of PowerPoint. Yeah. So we have unicellular trafficking, secretion, and vesicular transport going on in these yellow things, for example. 2 striking groups is this one over here. Function unknown is probably not entirely accurate, but that's the name that it's given in the database. It represents a large number of genes that they have been annotated, but there's no specific function you can assign all of those genes to. Here, general function prediction only, well, again, a function has been predicted, but it's not at a very specific level, such as those. Now, the shapes are indicative of the clustering method the layout method that I used, which is initially it's based on similarity, pairwise similarity between members of clusters. So if there is a similarity between a protein in cluster A and a protein in cluster B, I use the minimum such similarity as a similarity for the two clusters. If there isn't, I used a measure of the functional overlap, the extent to which they have the same function. So this lot here, these are characterized by having multiple functional annotations associated with them, and or also having, although they may be different functions, they have some kind of level of actual protein similarity between proteins in the two different groups. So that's how we get clearly defined groups, but then a sort of a mixing area in the centre. Okay, so to do the map layout, I resorted to Salmon's algorithm, first published in 1968. It's elegant, it's simple, and it's quadratic, and it's not really amenable to any kind of approach that uses second derivatives, because there's just so many weights involved. But anyway, using this, we got a reasonably efficient implementation of a steepest descent based algorithm, and laid them out so that, as far as possible, the distances in our layout resemble the arbitrarily high dimensional distances between the objects. So the distances are one dimensional, but the objects reside in an arbitrarily high dimensional space in the beginning.
I understand what you mean about the azimuthal distribution, but what do you mean about the radius? In other words, there's something farther from the center in the diagram you showed. What does that mean?
Well, okay, if they were all equally, if this collection consisted of 12 objects and they were equally separate from each other, they would tend to lay out onto the perimeter of a circle. So the things that are out on the edges of that circle are things that they're there because they're being as far away from everything else as they can be. And given we've only got 2 dimensions and we've got 25 clusters, the best they can do is sort of distribute themselves around the edges of the room.
Is it fair to say the measure how certain you are that they're different than the next cluster?
Well, they're different in the sense that there was no statistically significant sequence match between any protein in either of these remote clusters. So this basically means, okay, they're either infinitely distant or they're unknownly distant, but I wanted to get them into the map, so I allocated them a number, which infinity is probably 10 in this map. So if you make infinity smaller, then you get a much more compact clustering, and it tends to be try to distribute itself. But this gives an aesthetically pleasing result. We still seem to have a reasonable preservation of the similarity, and that's more important, is that similar clusters hopefully resides close together on that map. Okay, in our tool that we've produced, we can build 2D maps, 3D maps. We've tried to produce an immersive visual navigation metaphor, and we support multiple levels in this. Well, there's a 3-level hierarchy. We have the universe, and we have perhaps within one function, and then we have within one cluster. So these are natural artifacts of the underlying data set. Now we're building in some tools, some ability to filter it and to interconnect multiple views. As a case study, I took the COG database and then used BLAST to map proteins in two bacteria, Bacillus subtilis, which is a harmless soil bacterium, and anthrax. So these are Okay, there's 4,000 genes in Bacillus subtilis, 5,000 odd in anthrax. There's 192,000 proteins in the BLAST database. So what we end up with is several 100,000 hits when we do this on a genome-wide basis for each genome. It's not something you want to do every day on your laptop because it takes 2 hours and you can't do very much else while it's running. But at present, for the prototype, we've parsed it, we've loaded it into SQL Server, and we then process that. So, whereabouts do the proteins from Bacillus subtilis go? Well, that's the green highlights here. So they're sort of all over the place. Can we turn the lights down? Good. You can turn them.
I don't kill the power. Is that better?
Yeah. Maybe I should have chosen a different background colour too. I'll speak to my stylist about this. Okay, now on the next page. Okay, that was somewhat delayed. Where's my page up and page down? So that's anthrax. So if we sort of jiggle back and forwards, we can see that their composition is not identical, which is no surprise. I did a thing that an astronomer might enjoy, which is I created a blink animation so that we can visually detect differences between. This will eventually find its way into the tool as a Boolean operator where we can filter by membership of multiple or none or mutual exclusion type of relationships. Now, here's a picture of the proteins in one particular cluster. It's the biggest one. I chose it mainly because it is the biggest. And again, there's it's hard to make out, but there's some green dots amongst there. And in the tool, there's a capability to change the intensity. So now this is where it starts to get a bit shaky. All right. Thank you, Mike. Okay, so let's quickly find out. Okay, the data. One of these BLAST result data sets. This is it's either anthrax or Bacillus subtilis. Okay, it produces an XML document that's the better part of a gigabyte in size. What does the data look like? Well, it looks like this. So now, if we use one protein at a time, we can probably do something manually with this kind of data. Without automated help, we can't do anything better than we can't really do anything better than writing scripts and querying for very specific matching criteria. In particular, what we can't do with scripts is we cannot find things we didn't already know to look for. We won't be able to find emergent patterns by that. So I'm down to 2 minutes, so I will demonstrate running software, but it won't be big running software, it'll just be little. So I've loaded in a collection of hits from one COG. I can make this a bit bigger, and I can probably increase the viewing panel. So in this tool, we can use the mouse to move things around and see structure. Okay, we can, when we double click on a row, we can go and find out where that object is, and we can investigate a certain amount of information about it. This prototype is holding up better than I expected so far, actually. We've been live for over a minute. Now, we have the capability to have multiple maps. In particular, you can make a 2-dimensional map, a 3-dimensional map, and there's a couple of other parameters that you can use to characterize things. So we'll very quickly build a 2-dimensional map of that data. When I said quickly, it was a figure of speech. So this is it's a 2-dimensional display inside a three-dimensional rendering environment. So we can do that. We can save this map and users can reload it later, and we can finish our presentation because I think it's about time. So, and if I had one more minute, I would just run a movie that shows the formulation of the big cluster. Have I got 1 minute, Mike?
Yeah.
Okay, I'm sure there's a lot of MBF stuff in Windows Media Player, which... Okay, this is sped up by a factor of 6, but it shows the layout algorithm in action as we render those things. So that's everything I was going to show you today. I would like to close by expressing gratitude for to the workshop organisers for giving us the opportunity to present here today. And also I'd like to express gratitude to Microsoft External Research for providing funding that has assisted us in this research. Thank you.
Okay, we have time for a couple questions.
Hi, MC Shrey from the University of Southampton. Visualization is always really tricky. So I'm wondering, especially for large data sets like this, what made you decide on a particular visualization that you're using? And then how did you check that with the scientists to make sure that they were finding it meaningful and effective for their work?
I'm glad you asked me those questions. Well, firstly, a visualization, we're restricted to a small number of dimensions, 1 or 2, actually 2 or 3. You can project this into 1 dimension, but it's essentially useless. The salmon algorithm is appealing because it doesn't care where the data came from. It doesn't really care about the original dimensionality. We're not attempting to do clustering ourselves here, so we don't face that problem. This clustering is something that's inherent in the data that we started with. So the difficulties associated with assigning clusters to these mutually sort of incomparable things, that's been taken care of by somebody else. The biological significance in the underlying data is taken as a given because the COG dataset is used to classify proteins on a regular basis at NCBI. So the last of your questions, well, we do have graduate students who are looking at using this software in conjunction with in conjunction with regulatory network information or phylogenetic information to assist them in some of their research.
Forgive me, I think I wasn't clear then. I guess what I'd like to really understand is, have you found scientists are now able to remove things with this particular visualization that they haven't been able to do before because you've given them?
Well, not yet. This software is very new, and we're at the point of introducing it to collaborators within our university. So the proof of the pudding shall be in the eating, I think.
You mentioned that you were interested in interlinking the different displays, and that sounds great. And so I was curious, you just said something like phylogenetic information and other information, and so you presumably have that information about a lot of the same things.
Yes, we have that information. How it's going to pan out inside a visualization tool is an ongoing exercise that one of the other graduate students is dreaming up at the moment. But the other obvious thing that one can do, though, is one can link out from here to Entree and to Bio2RDF. We can and get to PubMed and pull together that kind of information.
Things that we like so much about Worldwide Telescope is the ability to link out to other programs. And what we're working on now is connecting it to tools, existing tools, and there are these hubs, resource hubs that can let the programs talk to each other. So I'm just curious.
No, we haven't gone that far. At this point, my presentation has been about the visual interface, the layout mechanism, and the proof of concept that it can even be done. The the prototype will be appearing, the source code will be in our CodePlex site sometime before the end of October. I'll just make it a little more stable. And then we'll be able to move on and interoperate with Excel, for example, so people can easily get stuff in and out of these systems, project it out to the web using Silverlight. So that means we have to do a bit of clever algorithmics to make sure that the right quantities of data go in the right place. But there's plenty of inspirational examples. The Worldwide Telescope, Deep Zoom, all of these technologies give us clear ideas about how we can go there.
Thank you very much.
Thank you very much.
Okay, everybody, thank you very much for coming to the second half of this part of the tutorial workshop. I'm happy to introduce Dennis Bromley from the University of Washington. He's going to be talking about DIVE, the data-intensive visualization engine. Please help me welcoming David to the event.
Thank you. So thank you very much for being here. We've been working in the DAGA lab at the University of Washington to build DIVE, which is a visualization engine and a data analytics engine. So why we built DIVE to begin with, most of the research in our lab deals with protein science, protein simulations, and drug design often. And so the challenges that we ran into, we have big data sets, which I'll talk about in a moment. We have complex data. Most of our data points have between 30 and 50 dimensions a piece. Different technologies and skills. Some people come in, they're hardcore Python people or C++ people or not programmers at all. And of course, we'd like to prevent a loss of knowledge as the lab turns over. We have graduate students and postdocs and people coming in and out, and we'd like to try and hold on to the work that they've done when they move on. So, so basically, often the way it works is we have data sources. Those get imported into a data model. The clients consume that data model and do some analysis. And this is really where we see DIVE playing a role here. And the data sources that we are dealing with primarily in the lab is we have the dynamiomics data set. Dynamiomics is a high-resolution set of atomic simulations. We're talking on the order of hundreds of terabytes in here. We should point out, though, that this is a plugin model to DIVE for the data source. So it would be fairly easy to write another data source that would pick up from flat files on a file system, Excel spreadsheets, anything like that, and including real-time sensors, which is sort of a fun thing that we're interested in exploring more. So the object model, so we have our data warehouse up here, and we bring a subset of that data that we're interested in into DIVE. And so the first thing we need to do here is need to sort of objectify that data. And we basically type it. We say, hey, this row of SQL data corresponds to an atom, and like atom number 16. OK, fine. And so once you do that, things get objectified pretty well. And so the SQL client now has full access not only to the original SQL data, but also the type and the object metadata as well. Similarly, the CLR client over here not only has access to all of that, but goes a step further and actually generates .NET objects on the fly that you can use at runtime when you're exploring your data. So the SQL client is primarily going to be used for large data sets. And the CLR client is primarily going to be used for visualization. Hopefully we'll get to a demo here at the end. And .NET interactivity. And this .NET interactivity basically comes in three flavors. The DIVE client is itself a .NET control, and so you can embed it into any other application and use those interfaces to drive it. You can also host it inside a scripting language. We've driven DIVE now from IronPython and F# both. So as long as you can host it in any of, basically our solution to the multiple scripting languages in the lab was to allow anybody to use their favorite one, and they can all speak to DIVE. And then embedded C#, we have a C# scripting window in the DIVE client, as well as inline C# microscripts, which I'll talk about in a moment. So analysis. Again, the SQL client is intended to do basically large-scale analysis. The CLR client is intended to do programmatic analysis. And one thing that we're also interested in looking at here are sort of bridging technologies, where SQL Server CLR down in the SQL engine could actually take advantage of the object technologies. Similarly, link to SQL could allow us to have some of the expressivity of SQL up in the CLR world. So that's something we're looking forward to doing too as well. So once you have these analysis, you can do these sort of iterative analysis where you take a piece of data, you do some analysis, you generate new data sets, and you write that back in a well-formatted way back into the database. When that happens, it's now formatted just like the other data, and so you can iterate on that and do analysis and cascade down. Which brings up the question of provenance, right? Anytime you do analysis to a data set, you'd like to know what happened. So in Dive, we have data level provenance. We know where the data came from, the original select statements, things like that, out of the data warehouse. We also know what was done to it, what analysis was done to it to create the new subsequent data sets. And then from that, we have basically a map. So if somebody's left sometime in the past, we have a way to recapitulate the data that they were working on. We also have workflow provenance baked into the CLR client. We basically start logging events. So as you're transforming things, as you're looking at things, we record that out, and we stamp it with time and date and user. It's basically a virtual lab notebook. And we also have a place where the user can enter their comments. Those get flushed out. So hopefully, sometime if somebody leaves the lab, sometime hence, you can go back and say, oh, he was interested in this. He had this hypothesis. He did these transforms to the data, and here's the figure that he published in the paper. And something that we're working on right now is to have the any analysis and data creation that happens in the CLR client propagate itself back to the object model back in the database. So the visualization engine is XNA. It's been very good to us. It's high performance. It's GPU accelerated. It's cross-platform. We're sort of interested in where we could go with phones and the Xbox and that kind of thing. However, the visualization engine is a plugin, just like the data source. And so we should be able to swap it out. There's been a lot of really good work in data visualization, and we'd love to be able to leverage that if we're looking for something and haven't found it yet. So the front-end client is basically where most people are going to be spending their time. And the role of it is to really take a raw property value, like one of those cells in the database, transform it, based on the transform value, filter it to decide whether or not it's something you're actually looking for, and then map it onto the axis. And so the cool part about this is that those two steps there are what we're calling C# microscripts. And so what a microscript really is it's a Lambda function under the hood. And so in the GUI, under transform function, somebody might just type X. What they're actually doing is they're completing the Lambda function, and then I compile it in memory, and then that is the transform that gets applied to the data. And similarly, if you'd like to filter it, you have to have a Lambda function here that returns some Boolean. If you just like to return everything, you can just type true, and we'll complete the Lambda function right there for you. So the result here is that even somebody who's not a programmer is able to use this. But as you can look further on, you can do arbitrary transforms. You want to have f of x as x squared. That's fine. If you'd actually like to only show the data points that line the main chain of the protein, you can just do dp.mainchain, and you'll only see those. So it's very straightforward. And of course, what's nice is that this is an arbitrary C# function. You can do arbitrary work in there. So you have all the .NET math functions, anything you want, any libraries you want to import. And this is all in the GUI, again. And you can do sort of arbitrary logical operations as well. A couple interesting things to call out. Here you can see that object model at play. Instead of typing dp.getPropertyValue, quote, main chain, all that stuff, we actually create these objects on the fly using the dynamic language runtime. And so you can just script it as if those data points and objects had come with those properties when they were born. Another interesting bit here is this line right here. You can do arbitrary transformations to the properties that are coming in, and including ignoring it, right? So if you have some external data source that's streaming live data, you can pump that in right here, and it'll just flow right into Dive, and Dive will start visualizing it, and you're good to go. So with that, I'd like to get to a demo. First, thank you to the organizers of this. Microsoft External Research has been great. Our lab back at UW, the NIH to NLM for a great deal of funding, the DOE Office of Biological Research, and the NERSC Supercomputing Center for helping us with the Dynamiomics project. So with that, we'll get to a demo. And do I still have time?
Yeah, you're doing great.
Sweet. Wow. All right. I got time for a demo. Okay, so this is Dive. It's the Dive client. This is not on the screen. Yeah, that's true. I had so much screen I had so much screen real estate, and I was kind of wondering what was going on. No, this is good. Okay, so let me expand this out a little bit. So this is the Dive client, and right now I've loaded the atoms and covalent bonds for a data set for the COMT, protein called COMT. It's responsible for interacting with various neurotransmitters in your body. And so the first step when you interact with Dive is you want to assign the basic axes. So one of the ideas of Dive is that you have a bunch of axes. You have X, Y, Z. For plotting things, you have color. You might have alpha. You have a few sort of different axes. And so you want to just do this mapping from the multidimensional data that you have onto those. So one of the most logical ones, of course, is to map the X, Y, Z axes to the X, Y, Z coordinates from the atomic simulation. And then we'll actually see. There we go. And then we'll actually see our molecule. And that's the molecule smashed into 2D. And once we pop out the z-axis, life will get more interesting. Okay. So there's COMT. There's our molecule. And let's see. And so one of the things we can do is the node color axis, right? So we can assign it to a property. And we could assign it to, say, residue ID. Residue is another name for just amino acids, for those of you who aren't familiar with that. So the residue number, and that basically shows us that we're looking blue, goes from blue to red. So here's the beginning of the protein chain. And it's going to run around and end up in the red zone there. This doesn't really tell us very much, however. So it might be more interesting, actually, to color it by SASA, which is the solvent accessible surface area. And so when you do that, let's see if I can pop this open a little bit more. You can actually see, again, running blue to red, low to high, that there's a blue core, which clearly the hydrophobic core of the protein is not being exposed to the solvent, and the stuff around the edges is exposed to more. We seem to be saturating a little bit on the color, so one of the things we can do is, let's see, let me make sure I'm on the right axis here. Hello, there we go. Yeah, so on the color axis, we can actually set a transform. And let me drag this over. And so instead of just plotting x as it came in, we can do math dot square root of x. Again, sort of performing arbitrary transforms on there. That's now compiled in memory, and that gives us a little better color separation. You can really see some of these side chains that are hanging out into the solvent. So this is great, but we also have additional axes, right? So one of the things we can do is we can look at, we have size, the size of those. So do I have a scroll bar? Give me 1 second there. There we go. And so I can size these by RMSF, and that's root mean squared fluctuation. It's basically how much the amino acid wiggles around in solution, or how stable it is. And so if we do, we'll just set the X, Y, and Z axis widths to RMSF. Now, there we go. What we can see is basically what we're looking at here is we still have the color to indicate that things approaching red and yellow and orange, that kind of thing, have a great deal more solvent accessibility. But you can also see here that the larger, are those skewed or are those spherical? Okay. The larger they are, basically, the more they wiggle around. So you can look at this. You can see a great deal of the dynamics inherent in the protein. And yeah. So there. And now, if you'd like to see just the main chain, for instance, we have the filtering capability. So we can come down. Let me go back over to the x-axis over here. And we can filter things. So now this is actually using the object model. So I can do dp.mainChain. And this actually used the dynamic language runtime and added these properties onto the object. So if I want to look at that, there's the main chain, just the carbon backbone of the protein. So you can look around at that. Now, this is, of course, over a dynamic simulation, right? So you can look at the different time steps and you can step through it. And then one of the other nice things here is that we can have integrated displays where they talk to each other. So I can load up, let's see, we'll load up atoms in this one. And if people are familiar with the Ramachandran plot, basically shows, so we're going to have the x-axis be phi, and this is basically showing the internal torsional angles of the protein. And y, that would be psi. And there are data points there. And might want to make those a little bit bigger. And so really the whole purpose of Dive, and there's a Ramachandran plot. And if we pull this out, let's see if this is going to lie here. If we pull this out, we can stack them next to each other. Maybe. I think we might be running into screen state. Oh, there we go. They're over there. Screen space. Will that work? Okay, well, there's we're going to play this way. So there's the backbone, and there's that. And as we step through here, you can probably see it just wiggling a little bit on the other side here. The Ramachandran plot. Woo. I'm not getting any love from this display here. That's awesome. Okay, so at any rate, the two are linked. So as you step through, the two will always keep in sync with each other. The idea is that you want to be able to show 5 or 6 dimensions in one view, 5 or 6 in another, 5 or 6 in another. And as you move through, everything will stay in sync. You can watch all of them together. So there you go. That's Dive. Thank you very much.
Very cool. All right. Questions?
Yes?
Having come from computational chemistry, and this is not, I'm not diminishing the work, but
No, please.
there are lots of programs that can do what you just demonstrated. So
Yes.
I'm just trying to I want to know what the value added from Dive is that I'm getting beyond, say, VMD, which can do all the stuff that you just showed.
Sure. So one of the first things is that this is not a molecular biology program. There's nothing about this. Dive doesn't intrinsically know anything about molecular biology. You can load up population data, traffic safety data, anything you want. Steve, sitting right behind you, is one of the co-authors on the paper, and we're both in the biomedical and health informatics department. So really, when we started building this, we wanted it to be relevant to our lab, but we also wanted it to be a general sort of informatics investigation tool. So that was sort of the original part for that. Does that answer your question a little bit?
Yeah. I just want to make sure that I got the point, or that was said. So that was, yeah. I thought that's what it was. But
Yeah, and also part of it is we were looking around, and we were on VMD and we were in PyMol and all those things in the lab, and they weren't, for the areas where we wanted to investigate, they weren't all sort of converging on the same area at the same time. And so we wanted something that was really we were setting out to sort of solve problems in our lab, and we saw this opportunity to solve this larger informatics kind of tool set. Anybody else? Anything? Sure.
I was just wondering if you could generate a 3D stereo of that images so that you can wear a Google a glass and see it.
We've actually talked about that. There'd be a couple ways. I'm sorry. Yeah, I should be repeating these. The question was, could we be generating 3D stereoscopic images? And we've actually talked a couple of ways of doing that. NVIDIA has an API for doing exactly that. And also, we could just make cross-eyed stereo or wall-eyed stereo views right in line. So there'd be a couple different ways to do that. Yeah. Yes?
You probably said it, but I missed it. What platform does this run on?
So this is all built in .NET. So at the moment, the platform is .NET speaking to SQL Server. But like I said, the SQL Server, if you wanted to come from a different data source, you just write a plugin for that. And it is running XNA for visualization. I suspect that most of the most of the rest of the client, aside from the visualization, could run in something like Mono. I don't know the capability of XNA running on other things. But again, the visualization is a plugin, so you could swap that out with Visit or any one of the other kind of data visualization tools you want to use. Yes?
What about publication quality graphics? Can you take a screenshot of this, or do you output a PRN file, or how do you get the interesting images into a publication?
Yeah, so we do. If you're in the screen, actually, Steve did a wonderful job. Steve actually wrote the renderer, as well as the database stuff. So yeah, it can save a PNG or JPEG or whatever you want out straight from there. We're also interested in pursuing some of the high quality shaders involved because it's an XNA. It's basically a video game engine, so you can play all those tricks too. Yes.
What's the largest size data set that you can use and still manipulate in real time?
So we've run millions of points in any given frame using this rendering situation. The per frame data set are actually streamed up on demand. I'm not holding them in memory. So we'd like to accomplish some. The test will be when we can visualize and interact with some data set that's considerably larger than the amount of RAM we have in the machine. And we're getting ready to approach that here. Yes?
I'm wondering if you just tell us a little bit more of the history of, you said that Kymall didn't do everything you wanted, and then there's other sort of famous statistical graphics programs that do these kind of things like Datasets. And so how did this come to be? I mean, you keep saying the lab, and I
Oh, sure.
kind of like who the people are and how many people are involved and what they were complaining about, like the sociology of the story.
Sure. So we're in Valerie Daggett's molecular dynamics lab at UW. And most of the work in the lab is done using these molecular dynamics trajectories. And so we have on the order of maybe 15 people in the lab right now. So lots of people coming from a scripting background, several people who are straight, I'll call them straight scientists, who have very little interest in the actual informatics technology of stuff. A lot of those people would want a very GUI-driven or point-and-click kind of interface. Then we have other people who are really interested in doing data mining, like Steve and myself are interested in the informatics side of things as well as the data mining. I'm interested in doing drug design, this stuff. So we wanted something that would bring a certain amount of coherence to all the tools that were in the lab. And so we kind of went down this road because this seems to hit all the big items that people were interested in doing. There's visualization, there's analysis, there's we really wanted to hit having multiple multi-dimensional displays so you can walk through things. And then I wanted to be able to satisfy the biologists and chemists who just wanted to basically point and click. I mean, to their credit, they're totally heads down in chemistry, so they're awesome. But at the same time, I didn't want to give up the ability to have this be really extensible. So that's why we went down the microscripting route. So that's a little bit of the sociology. I'm not sure I answered all of your question, though. Are there? Okay. Anybody? We're happy to give live demos after this. Thank you very much.